This is our third class on molecular basis of inheritance. Before starting today's class, you need to revise your knowledge on two topics. Number one, the composition and the structure of DNA and RNA, and number two, organization of DNA into chromosomes. In this particular class, we will discuss how DNA replicates. That means how DNA makes exact their own copies. As we know that Watson and Crick proposed their DNA double helix model in 1953. They also proposed a model for DNA replication, that is semi-conservative model. At that time, there were two other models. Number two, conservative model, and number three is dispersive model. According to this model, this conservative model, DNA would get copied and this will make a new DNA. Suppose this is a DNA molecule. It will get copied. That means it is the copied DNA and it will produce a new one, that is the new one. The black one is the new one. This is the conservative model. And now dispersive. According to this model, according to this dispersive model, DNA would get cut. DNA would get cut. That means this, many cut. And each of them will produce a new copy. That is and then they will get attached. That means this will happen. Now the semi-conservative method. According to this model, DNA, the two strands of a DNA would get separated. That means the separated DNA, one strand is this and one strand is this. And each of them will produce a new one. That is this one and this one. So these are the models for DNA replication. All of them were hypotheses. This semi-conservative model was proposed by Watson and Crick, and this conservative and dispersive model was proposed by other scientists. And that time, most of the scientists believed that the two strands of a DNA could not be separated. In 1958, Meselson and Stahl designed an experiment to know the actual model of DNA replication. Now we will discuss their experiment. In their experiment, Meselson and Stahl used E. coli bacteria. As the DNA has nitrogen in its base, so Meselson and Stahl grew this E. coli into a medium containing ammonium chloride, and this N is the source of the nitrogen of this DNA. But this N is not normal. This ammonium chloride is not the normal ammonium chloride because it has 15 isotope. The normal nitrogen is N14, but N15 is the heavy isotope of nitrogen. It is not the radioactive isotope, it is the heavy isotope, as it has an extra neutron in its nucleus. So when the E. coli reproduce in this medium, they use this N15 as the source of nitrogen, and as they reproduce, they actually replicate their DNA. So in this process, they use N15. This is the first step. Next, they transferred this bacteria into a culture medium containing N14 ammonium chloride. This is the normal ammonium chloride, and let them to reproduce. So when they reproduce, that means when they replicate their DNA, they use N14 in this case, and now they collected the bacteria and extracted their DNAs and dissolved the DNA into a solution of cesium chloride. So this is the first step, where the bacteria grew in N15 solution. Then the second step, transfer into N14 solution, N14 ammonium chloride solution, and then they dissolve this DNA into cesium chloride. Then in next step, they centrifuged it. That means centrifugation. As we know that after centrifugation, the particles of a solution get separated according to their weights. So when they centrifuge this DNA contained solution, that means the sample solution is spun rapidly in a centrifuge with high speed. So as a result, they found the separated particles. So in the test tube, after centrifugation, N15 will be settled down as it is heavy, and they produce band here in the bottom, and N14 will be on the top, and they produce band here. This is the setup of their experiment. Now it is the time to observe their result. After the first generation, when they centrifuged their solution, they found that the band is neither on the top or in the bottom. They found the band on in this intermediate part. This is on F1 generation, and in F2 generation they found a band on this intermediate part and in this upper part. That means this band is for N14 and this band is for the mixture, the intermediate part. That means it has N14 and N15. This is also N14 and N15, but there is no band for N15. This is their observation. Now it is the time to interpret their observation. So when they grew the bacteria into this N15 solution, all the DNA strand of this bacteria will be N15, And if the conservative method was correct, so they will produce exact copy of this DNA, that means N15 N15, and this will produce a new DNA that will use this N14, this new DNA 1414. So if the conservative method was correct, we would get a band for this on the top position and a band for this on the bottom position, just like that. But Meselson and Stahl did not get it. So the conservative method was not correct. Now it is the time to check the dispersive model. Just like the previous one, as it is grown in 15 solution, both the strand of this DNA will be N15 and 15, and as we discussed earlier, they would get cut just like that. This blue is for N15, and they will synthesize a new one, this, just like that. This black is for N14. So we will get an intermediate band, but Meselson and Stahl did not get it. So the dispersive method is also rejected. Now it is the time for checking the semi-conservative method. Just like the previous one, both the strand of this DNA is N15 and 15, and according to semi-conservative method, both strands will be separated, that means this, and they will produce a new one. This is for F1 generation. So in F1 generation we will get an intermediate band, just like that. And for F2 generation, again they will get separated. So this is for the N14 and this is for N15, and they will produce a new one. New one is always N14, so they will produce this, and this new one will be obviously N14. So this will be N1414 and this will be N1514. So the 50% DNA will be 1415 DNA and 50% will be 1414 DNA. So there should not be a band for N15 region. So we will get a band on intermediate portion and on the top portion, just like that. So now it is clear that the semi-conservative method for DNA replication is correct. Meselson and Stahl concluded that. In our next class, we will discuss the process of DNA replication. Now the question is, what kind of isotope did Meselson and Stahl use in their experiment? Was it radioisotope? Comment your answer.